Chapter 5, Microbial Metabolism. In this chapter, we're going to be covering reactions underlying metabolism, carbohydrate catabolism, other catabolic pathways, photosynthesis, other anabolic pathways, integration and regulation of metabolic functions. So I'm going to be covering reactions underlying metabolism because this overview is very short. And then we'll hit each of these others separately, except for other anabolic pathways and integration and regulation of metabolic functions we'll cover together. So let's begin the first topic, basic chemical reactions underlying metabolism. The learning objectives for this topic are distinguish among metabolism, anabolism, and catabolism, contrast reduction and oxidation reactions, compare and contrast the three types of ATP phosphorylation, and make a table listing the 6 basic types of enzymes, their activities, and an example of each. Describe the components of a holoenzyme and contrast protein and RNA enzymes. Define activation energy, enzyme, apoenzyme, cofactor, coenzyme, active site, and substrate, and describe their roles in enzyme activity. Describe how temperature, pH, substrate concentration, and competitive and non-competitive inhibition affect enzyme activity. So here are the topics that we're going to cover under this topic: catabolism and anabolism, oxidation and reduction reactions, ATP production and energy storage, and roles of enzymes. First, let's define some terms: catabolism and anabolism. Catabolism refers to the reactions within a cell that are the taking large molecules and breaking them down into smaller molecules and releasing energy in the process. So if you think back to chemistry, these would be exergonic reactions, reactions in which the reactants have more energy than the products do, and there's energy released. Now, anabolism refers to reactions within the cell that take smaller precursor molecules and, using energy, build larger molecules. Now, this would be endergonic reactions, or reactions in which the reactants start out with less energy than the products, and energy is used in the process of the reaction. Taken together, the catabolic reactions and the anabolic reactions together are the metabolism of the cell. Let's walk through how this works for the cell. Now you'll notice that there's energy lost as heat during catabolic reaction and anabolic reactions. Whenever you have a change, a chemical reaction, you're going to lose some energy as heat. That's the same principle that applies. You can't take the exhaust from your car and run it back into your fuel tank and expect the car to run. Just like that, the cell can't take the waste, the heat that is lost, that is unusable to the cell, and use it to continue to run the cell. The cell constantly has to bring in some sort of energy. In this case, nutrients. So these nutrients are used to generate the energy that enables the cell to build its macromolecules: carbohydrates, lipids, proteins, and nucleic acids. Now all of these macromolecules go on to make cellular structures, to be a part of cellular process such as cell growth and cell division. So we've got this cell is starting to divide. And it's also energy storage that can go back into catabolism. The reason why you got to have energy storage in the form of macromolecules is that ATP doesn't last very long. If you don't use it in an anabolic process, it will just naturally break down in ADP. So you have to use it as soon as you make it. Now we're going to talk about oxidation and reduction reactions as it applies to cells. Now, the reason why we bring this up is because oxidation and reduction reactions happen all the time in metabolic pathways. So it's good to know what's happening. So first off, let's talk about these electrons. Electrons carry energy with them. So when I move this electron from this molecule to this molecule, energy is going with it. Now, when we have the electron moving from this electron donor to an electron acceptor, the donor is oxidized and the electron acceptor is reduced. Let's look at an example of a set of oxidation reactions or redox reactions as seen in metabolism. So here's a reaction that you're going to be taking a look at in the next topic, in which pyruvic acid loses this CO2 group and we end up with acetate. Now, this acetate, the hydrogen here, is moved to NAD+. Now, I said electrons carry energy. Sometimes the proton goes with it, and it's the entire hydrogen. So whether you're moving an electron or moving a hydrogen, energy goes wherever the hydrogen or the electron goes. So in this case, we're moving the entire hydrogen, and it's moving from acetate to NAD+. Then we get NADH, and then we have a coenzyme come on, and we get acetyl coenzyme A. So, which one is oxidized? Well, the acetate is oxidized. It is losing that hydrogen and the electron that goes with the hydrogen, and NAD plus is being reduced to NADH. So, you're probably wondering, why do I have to learn which one's being oxidized and which one's being reduced? Because you need to follow the electron, because that's where the energy is going. NADH has more energy than NAD plus because it has that hydrogen. Now, we've lost some energy from the acetate. We still have plenty of energy in acetyl-CoA, and we'll learn more about that in the next topic. But energy has been transferred to the NADH. Now, later on, this NADH can transfer a hydrogen. It'll be oxidized, go back to NAD+. It'll donate that hydrogen and donate the energy from that hydrogen to another molecule or another enzyme in the process of generating more energy. Okay, now you're probably going, as I did at this point in my college career, I'm never going to remember which one's oxidation and which one is reduction. So I have some mnemonics to help you. I have two of them. Here's my favorite. Leo goes grr. Losing electron is oxidation. Gaining electron is reduction. See, Leo goes grr. So if you're losing an electron, you're being oxidized. If you're gaining an electron, you're being reduced. Now, lots of my students prefer oil rig. Oxidation is losing. Reduction is gaining. So whichever one suits your fancy, Leo goes grr or oil rig, you can pick which one works to help you to remember the difference between oxidation and reduction. Let's talk about the other major way that cells transfer energy, and that is through ATP. Here is the structure to remind you what adenosine triphosphate looks like. It's where you have an adenosine that has 3 phosphate groups. Now, when you clip off one of these phosphate groups, you get adenosine diphosphate, or ADP. And then if you clip off another one, you get adenosine monophosphate, or AMP. Now, for each loss of a phosphate, you lose energy. This is kind of like with the redox reactions, with the energy going with the electron or the hydrogen. In this case, the energy goes with the phosphate group. So the fewer phosphate groups you have, the less energy you have. So let's talk about these types of reactions. Now, the first one is substrate level phosphorylation. I know that sounds like a whole bunch of Greek. Actually, it's Latin. Phosphorylation is the process of putting a phosphate group on a molecule. So if you are phosphorylating something, it's getting a phosphate group. If you're dephosphorylating, you're removing it, and something else is getting phosphorylated. So what do we mean by substrate level? Now remember, the substrate is what an enzyme acts on. So all substrate level phosphorylation has an enzyme involved. So let's take an example from glycolysis, which we're going to be talking about in the next topic, where we take glucose and we take ATP and we move a phosphate group from ATP to the glucose, just like that. So the glucose has been phosphorylated, and it is a substrate. An enzyme has taken the phosphate group from ATP and moved it to the glucose, and you end up with glucose, 6-phosphate, and ADP. So anytime you have phosphate groups being moved between one molecule to another, that is substrate level phosphorylation. Now we have another one. This one is called oxidative phosphorylation. That's where you have phosphorylation happening in the process of consuming oxygen. Now we're going to get more into that later, but let's take a look at what the end reaction is. That's where you take ADP, an inorganic phosphate, and through the action of an enzyme and without another molecule being involved, you smush them together to get ATP. So there's no substrate except for the ADP and the inorganic phosphate. And oxygen is consumed, so we call it oxidative phosphorylation. Your cells are doing this even as we speak. Now we have another one called photophosphorylation. It's pretty much the same as oxidative phosphorylation, but instead of consuming oxygen, we use light energy to accomplish this phosphorylation of ADP to ATP. In this chapter, we will be looking at all three of these types of phosphorylations and how ATP is either consumed or produced. Now in our last subtopic, we have three subtopics. In other words, when we're talking about the roles of enzymes, it's good if we break it down. So first we're going to talk about naming and classifying enzymes. We're going to talk about the makeup of enzymes, and then we're going to talk about enzyme activity, including regulation of enzymes. So before we get into the role of enzymes, what are enzymes? Enzymes are proteins that act as catalysts in biological reactions. So what is a catalyst? A catalyst is anything that speeds up a reaction or is involved in a reaction without being consumed by the reaction. So in other words, we can take enzymes and we can use them over and over and over again. Whereas the substrate for the enzyme is changed during the reaction. So now let's talk about naming them. Most enzymes end in ase. So if you see an ase, like sucrase, that would be an enzyme that has something to do with sucrose. Specifically, it breaks sucrose. So let's take a look at the different types of enzymes. So you have hydrolases. They break molecules by adding water through hydrolysis reactions. They're also called hydrolytic enzymes. There are lyases. They split molecules without the addition of water. Isomerases rearrange atoms within a molecule. So they take a hydrogen and they switch it with a hydroxyl group or vice versa. They do all sorts of fun stuff. Polymerases join two molecules. They're also called ligases. So if I'm making a protein, I'm going to be using a polymerase that the substrate is amino acids. Okay, oxidoreductases. I wasn't quite sure I was going to be able to say that. These are enzymes that participate in redox reactions, or the movement of either a hydrogen or an electron between two molecules. Transferases transfer functional groups between atoms. So functional groups would be things like a methyl group. A hot topic nowadays is epigenetics. And that's where you have environmentally influenced changes in gene expression. And a lot of this comes from a specific type of transferase that moves methyl groups onto DNA to either turn on or turn off genes. Isn't that cool? And then we have ribozymes. Ribozymes are kind of cool because they're not really enzymes. They act as enzymes, but they're not made of proteins. They're made of RNA. Up until fairly recently, I would say within the last 40 to 60 years, in science that's fairly recently, we did not know that there were any enzymes that were anything other than proteins. Well, we started finding that RNA had some enzymatic functions. Well, since we'd already defined enzymes as being protein catalysts, we had to come up with a new name, and we came up with ribozymes. There are ribozymes that are in eukaryotic nuclei. They're the ones that do the splicing of eukaryotic messenger RNAs, removing the introns. You also find ribozymes in ribosomes. Isn't that appropriate? Enzymes often come in sections. So the main part of the enzyme is what's called the apoenzyme, and it's made up of a protein, but it can't function as an enzyme because it's not complete. It needs to have cofactors. Now, cofactors can be either inorganic. This is probably an atom of iron or magnesium or mobilium or something like that. And if you have something that is organic but not a protein that is important to the functioning of the enzyme, we call this a coenzyme. A coenzyme is an organic cofactor, whereas oftentimes if we say just cofactor, we're referring to the inorganic cofactor. Now in this particular enzyme, once we put the cofactor in and the coenzyme into the apoenzyme, we get the hollow or whole enzyme, and we have an active enzyme. Now this spot here is called the catalytic site or the active site. So on this diagram here, we have the enzyme, and this one has two active sites. The substrate fits into it perfectly, and the active site does whatever the enzyme is going to do to the substrate. So you can think of the rest of the enzyme as just getting the shape of the active site.